going to draw them the way I did the other day too, which is to say that this is a cell that's been infected, and what it's going to do is take a little piece of that bacteria, bind it to a protein called MHC, stick it out on its cell surface, kind of like a flag to say, hey, I'm infected. So this right here is the MHC1 on all cells with a nucleus. Yes, sir.
Does it only work with bacteria or viruses too?
Viruses too, any kind of pathogen. When this TC cell finds an infected cell and it's got this connection, it's going to do two things. It's going to make memory. We're going to come back to MHC in a second, but I just wanted to follow through on this one. It's also going to make more active cells. So you'll get additional TC cells made. And those will go back out and look for infected cells. And when it finds them, it's going to use a triple drug cocktail. It's going to do three things. It's going to disrupt the metabolism, so TC cells kill. By disrupting metabolism. It's going to make it hard for that sick cell to make ATP and energy anymore. It's going to stimulate apoptosis, APOP, QSIS, which is basically a set of genes that, when activated, will cause a cell to just die. So it's kind of a self-destructive type of button. The third thing it'll do is release perforins. We talked about perforins before when we talked about natural killer cells. So little proteins that will assemble into a ring shape in the wall of a pathogen with a hole in it. So this is how the TC cell kills. So out on the surface, this is a figure from a different textbook. Out on the cell surface, this is a virus, and this is a bacteria. This cell, this infected cell, is going to be able to get a piece of that virus or a piece of that bacteria and join it with its MHC1 protein. It's going to take that MHC protein, send it through the Golgi, transport it out to the cell surface, and put it on the cell surface as sort of a flag to say, hey, this is what's infected me. When it does that, in this figure, this is the TC cell, or the CD8 cell, and this is the infected cell. And this inset over here is showing the MHC1 from the infected cell coming together with the TC cell. When that happens, the TC cell will make additional memory cells. These are not going to fight the fight today, but they're going to be there the next time this pathogen is seen by this immune system. That's going to make more active cells. They're going to go out and find more sick cells. It's going to do three things. Release lymphotoxin, which disrupts metabolism. Release cytokines, which stimulate apoptosis. And release perforin, which is going to cause cell lysis. So essentially, those three things are going to cause the target cell to be lynched, to die.